microfilaments, desmosomes are stronger. That's the reason why desmosomes are stronger than the tight junction. When we say tight junction, we are referring to the point of connection. It is tightly connecting one with another. It's not mean strongly connecting one cell with another. Tight junction is connecting the weaker cytoskeleton, whereas desmosomes are connecting the stronger cytoskeleton. As if, just think, if your bones are connected, this would be more stronger than connecting cartilages. Cartilages are weaker than bone. This could be a good example. So saying this, now let's begin our next topic, which is glandular epithelium. Glandular epithelium epithelia are again various types of secretory glandular tissue we have in our body. So we need to classify them. We need to have a manner of identifying one from the other. And because of this, we have two major categories. One group of glands are releasing their secretory product by duct, or just they are releasing to a point from where they developed, which is surface or of the body or the cavity of luminal organ. We call this type of glands as exocrine glands, whereas those which are releasing their secretory product into the interstitium from where the product would reach by either diffusion or blood vascular system, we name this endocrine glands. Otherwise, both are developed from epithelium, covering or lining epithelium. The reason why glands are also considered to be epithelium is that they are all similar. The glands develop from the lining epithelium or covering epithelium. Look at this. This is a lining or covering epithelium. It could be your skin, or it could be epithelial covering of your oral cavity for development of the salivary glands. If it is skin, for example, for development of sweat glands or any other endocrine glands. These glands begin to develop as projections out. You see, cells multiply and they project deep into this underlying connective tissue. And then from here, the distal part would turn up to be secretory. They produce a substance, and the upper proximal part would act like a conducting tube. And the secretory products coming from the secretory portion therefore drains onto the surface of the epithelium. This type of glands have retained their duct. They are one or the major type of exocrine gland. This type of gland could also lose their connection like this. Their connection is now lost. The secretory índices are producing their secretory product into the interstitial tissue. And from here, the secretory product can reach to the target by diffusion, secretory substance, or need to get into blood vascular. sorry, we have under exocrine glands, one cell acting as gland, we name it unicellular gland. Sorry, let me first define the criteria for identifying exocrine glands. One is by number of cells, another one is by nature of their secretory product, another one could be based on mechanism of releasing their secretory product. Based on number of cells, you can have unicellular gland, like this one, where you have only one cell is involved as a gland, like this. Good example we have in our body are named as goblet cells. They look like goblet glass, where the secretory product is to be accumulated in apical aspects known as theca, whereas the organelles and nucleus are all pushed to the bottom. We name it as a neck region. And these are mucus producing type of gland. They are found isolated, only one. Usually they are coming in respiratory tract, GIT, along the other non-secretory lining epithelial cells, like this one, 1, 2. We name them goblet cells. One cell occurring as secretory, unicellular gland. Then the large majorities are coming as multicellular, and they come in different varieties. I will show you what we mean. This multicellular glands could be either as epithelial secretory sheet, intraepithelial glands, or the rest. The epithelial secretory sheet are glandular cells which are also acting as lining epithelium. They are lining epithelium at the same time producing substance as glands. And the whole covering or lining epithelium is gland. We call it epithelial secretory sheet. A good example we have is the stomach. You know, stomach is to carry on its digestion under acidic environment. And this acid could damage the cells themselves and then get down deeper to damage the deep tissues, connective tissue, blood vessels, like in condition of serious gastritis. And because of this, the cells need to protect themselves by their own secretory product, which covers the apical aspect of the whole cell. So every cell is engaged also in secretion. We call it epithelial secretory sheet. Another glands may be found in a given covering epithelium as an isolated cluster of few cells, like this. Here you have non-secretory cell, but this group of 4 of them are secretory. We name them intraepithelial glands. Intra is referring within. So these are found within the covering epithelium, but they are glands. And the rest of exocrine glands are widespread. They have their own duct system, and they need to be classified. And for such a classification, we have 2 important criteria. One is based on the nature of the duct, meaning whether it is one single unbranched, we name it simple gland, and when it is branched, we name it compound. So glands with ducts could be either simple or compound. Then the shape of the secretory endpiece, which may be like a tube, we name it tubular, which may be round, we name it acinar. It looks like a grape. When these acinar glands are big enough, looking like alveolus, we name them alveolar or saccular. So you see, based on shape, you can have 2 basic types of glands, either tubular or acinar, or even mixed. So we can have such glands under exocrine gland. Diagrammatically, you can see simple glands could be like this. Mind you, the blue outline is to mark the duct. The brownish one is the secretory endpiece. Because the duct system is not branched, both of these are simple, but this one and this one are different, not based on their duct system, but the secretory indipiece is branched. So it is not a compound. We call this one branched alveolar or branched tubular type of simple gland. So both of these are simple. These are different from this gland, where the duct system is divided. We name it compound. Such compound glands are big. Think of them like an orange. This orange is an organ which is covered by a capsule. Glands are also covered by connective tissue. We name it capsule. And from this capsule, partitions run deep inside. This capsule and the partitions are not epithelial tissue, they are connective tissue. And the partition deep running in the gland are named as septum or septa. The septa or septae would divide the glands into lobes and lobules, just like the orange is divided into compartments. Big compartment is named lobe, tiny ones are lobules. So glands, compound glands are also divided into lobes and lobules. Anything that is found within lobe or lobules are named as intra. Those found between them are inter. Septae are interlobular septa, interlobular septa. And from this connective tissue, tiny extracellular substance, fibrous type of extensions of the septae would run and support the epithelium. The epithelial cells are those which are principal cells. The capsule, septae, the others, non-epithelial cells are supportive structures. So we can divide them as stroma, which are supportive tissue, and parenchyma, which are the epithelial cells, which are the principal cells of that given gland. So such compound glands are therefore divided into stroma and parenchyma by the septae and the trabeculae into lobes and lobules, and then supportive tissues. They look like this. Look at this. This gland has lobe. You can call this one the big one lobe, another one, right, left lobe, upper, down lobes. And each lobe is divided into such lobules. 1, 2, 3. Tiny ones are lobules. And then look at where secretion is to take place. Secretion is to take place deep in the lobules, from secretory indipiece, to be collected by tiny ducts. The smallest ducts are joining one with another and running out towards the connective tissue partitions. And they are growing more and more, and ultimately they make the excretory or main duct system. The tiny ducts are lined by squamous epithelium. As they are becoming bigger, they will be lined by cuboidal or low columnar type of epithelium. And the main excretory ducts are usually lined by stratified type of epithelium. Based on the secretory piece, you can have tubular gland, acinar, or mixed. I told you. The tubular glands look like this. These are all simple. You see, simple tubular glands. These ones are straight. These ones are branched tubular. These ones are coiled. You can have different types of such glands. The acinars look like this one. When they are small, just call them acinar. When they are big, you can call them alveolar or circular. Most commonly occurring glands look like this. You read this, simple, and these ones are compound. Based on the nature of secretory product, the secretory product of glands could be either mucus or serous. Mucus is thick, serous is thick. Mucus is same as the mucus you see coming from your nasal cavity, for example. And it is thick. And this thick secretion needs to be produced by a secretory indipiece which has wider lumen. Such glands are usually tubular. So mucus secreting cells or mucus glands are usually tubular, whereas serous secretion is very thin. The good example I can give you as serous secretion is tear coming from your eyes. And this do not need wider lumen. So serous ones are usually coming as acinar. Mucus looks like this. Mucus looks like this. Mucus glands look like this with wide lumen. Serous looks like this with tight lumen. Sometimes glands may be mixed. One region serous, another region mucus, or they may be closely linked. When they are closely linked, after tissue preparation, you would see the mucus ones are found deeper and the serous ones are found peripherally. And by looking at this type of outline, people thought that originally, they logically thought that this type of serous gland is located peripherally as demilions, as half-moon shaped structure, which is named as serous demilion of 𝒊𝒔𝒕𝒐𝒍𝒐𝒈𝒚, is thought to be strategical because the mucus cells are located deep centrally, produce thick secretion, which requires thin secretion coming from the peripherally located serous cells, and this would dilute the secretory product, and the secretory product, the mixed one, is now relatively thinner, so that flow is facilitated. So this was thought the reason why you see in histology lab, serous found as serous demilion. But recent studies disproved this. Instead, they occur intercalated, mixed like this. Look at the serous and the mixed are very much closely intermixed. But during tissue preparation, the mucus cells would swell, whereas the serous ones would be pushed peripherally. So this type of orientation is concluded to be an artifact. It is an artifact, but logically was described to be reasonable. Therefore, it is not, this one does not exist in reality. Serous demilions are not existing in life, but they are found intermixed. So the reason why they appear later is because of tissue preparation. Then based on mechanism of secretion, you can have glandular cells producing by exocytosis. Name this type of secretory cells, merocrine, epicrine, or ecrine. Some other cells would accumulate their secretory product as the apical aspect. When they are stimulated to release, the apical part of the cell would be pinched up. So during secretion, there would be also loss of a small part of the apical part of the cell. This type of glandular cells are named as epocryne. Third type of gland is produced by rupturing. When they are full of secretion, they would accumulate it. Gradually, the cells would swell down up, and when they are needed to release, they rupture. And this type of glands are named as holocrine. Then we have another group of glands are endocrine. These endocrine glands do not have duct, so their secretory product named as hormone need to be reached. It is target either by diffusion or gets through blood vascular system. And those big ones usually are found associated closely with blood capillaries. This is because they need the blood capillaries not only for their metabolic demand, but also for delivering their secretory product. And they are found either as cords around blood capillaries, either as clusters or ball-like follicles. That's how they are found. Endocrine glandular cells could be autocrine or paracrine. They are named autocrine if the secretory product influences the cell itself. So the cells are producing secretion, and this secretion would also have the target, the cell itself. You may ask, why do they have the target? The cell itself is for maybe for feedback regulatory type of mechanism. When the target cell is neighboring, name it paracrine. So endocrine glands could be autocrine, paracrine, or have their targets located far away. And these far away located targets obviously would be reached by blood vascular system, and most are like this. And then finally, in haste, I can tell you one group of endocrine glands are found diffused throughout your body at different locations. And this type of glands are named as the diffuse neuroendocrine system. The diffuse is coming because of their diffuse location. The neuro is because their secretory product, the hormone, is also same as like neurotransmitters produced by neurons. So this type of glandular cells are like neurons. We name them neuroendocrine system. And this type of glands are again widespread. They are found isolated or in a small number, 2-3 cells isolated like these ones. They are found dispersed among other non-endocrine type of glands. This type of glandular cells are also named by different names. Some are named the epithelium cells or epithelium system by taking the acronyms from their behavior, because this type of diffuse neuroendocrine cells are able to take up amine precursor and decarboxylate it. And because they are named as amine precursor uptake and decarboxylating type of cells, they are named as APUD system or cells. Another group of such diffuse neuroendocrine glands are named as argentaphil cells or argyrophil cells because they have high affinity to be labeled by silver salts. Silver salts, chrome salts, and others are special type of staining procedures. So in this type of special staining procedure, the neuroendocrine type of cells are highly stainable. So because of this, they are named as argyrophil or argentaphil cells. Another group of cells are stainable by chromic salts. They are named as chrome salts. Adrenal medulla, for example, is famous to be stained by this type of staining. So the cells themselves are named as chromaffin cells. Another tissues of related cells which are stained by chrome salts are named as chromaffin tissue. And another group of such diffuse neuroendocrine system or glands are found in large numbers throughout our digestive tract, especially beginning from the stomach. They are found in small intestine, in large intestine, including the pancreas. So they are named as gastroenteropancreatic endocrine cells or enteropancreatic endocrine cells. The intro is coming from the enteric system. Enteric is referring to the gut. Pancreatic is because they are also found in large number within the pancreas, including the isolates of Langerhans. Those which are known to produce, for example, the insulin belong to this category. So they are also named as what? As gastroenteropancreatic endocrine cells. And because of their location in the gastric system. So that's what I can tell you about the endocrine system. And I think this will be our last slide. And with this, I've finished my lecture. I will come to see you after next week for another series of lecture. I hope that time we will not have this type of disturbance. Otherwise, I've finished my presentation from such few slides. I will send you a little bit extended version of my today's lecture as PDF version of a PowerPoint presentation. So you read that one and enrich it. If you have any question, feel free to ask me, especially when we meet for my next lecture. With this, I have finished my presentation. Are you with me? And do